So today we are going to have your presentation about certain physiological phenomena and the related biosignal that can be recorded to estimate or to describe that phenomena. So who should start first? Yeah, let's see. Let's give a toss with random number generator, whether it will be group 1 first or group 2 first. Wait. Random picker. Okay, group 2 first will is on this. Group 2, I just do some manual. Group 2 first will present. So Angeli and Grace will present their retinal activity measurement. So please turn on your video and you may start presenting whenever.
Disabled, sir.
Yeah, I just enabled it.
Okay. Kelihatan kan?
Yes.
Okay.
So I will start the presentation. Good morning, everyone. Good morning, sir. Today, Tanita and I will be presenting about electroretinography or ERG. ERG is a diagnostic tool that measures the electrical activity of the neural and non-neuronal cells in the retina as a response to light stimulus given. The tools for ERG is there is the electrodes as the sensor. The electrodes can either be the speculum shape, the cottonwood shape, or there is also corneal electrodes, which is inserted between the lower lid and the cornea, like the picture. Next. So the biosignals in ERG is produced by different cells in the retina. Here is the picture of the cells in retina. There is the pigment epithelium, roots and cone cell, Muller cells, on bipolar cells, of bipolar cells, and hemacrine cells. Each of these cells gives off different wave types. There are A wave, B wave, C wave, D wave, and also oscillating potential. This is the example of the graph that is produced when the biosignals are recorded. The A wave and the B wave are the most important ones because the other type of waves are sometimes seen and sometimes not. So the only ones used for clinical diagnosis is mainly just the A and B wave. Yeah, and this one, the picture in the middle, is also an example of the response of the retina when the light is turned on. The A wave you can see is in the front; it is in the negative amplitude, and there's the B wave and the C and D wave. Next.
Okay, I will be talking about the interpretation of the ERG itself. So, as we have known earlier that the ERG measured the electrical activity of our retina. So, if there is something in the retina that doesn't function well, it could be shown in this test. So, if for example we have a certain disease like diabetic, yeah, there will be a change of chemicals or something like that in the retina, and it could be indicate using this type of test. So an abnormal ERG means that more than 20% of the retina is affected. And non-dilating pupils affect ERG. So that's why before the test, I think the doctor will drop an eye drop or something like that, so that our pupils could dilate, so that the stimulus can go through to the retina. And here are the types of abnormal ERG. First is the subnormal ERG. It has a potential of less than 0.08 millivolts, and it means that the large area of retina doesn't function. Some example of cases are early case of retinitis pigmentosa, retinal detachment, anemia, and others. Second is the negative response. As we have seen earlier that the graph should be the negative will not have this large amplitude, but if it has this large amplitude, and that it means that there is a disturbance in retinal circulation. And some of the cases are cancer-associated retinopathy, siderosis bulbi, melanoma-associated retinopathy, and others. And this is the third type of response, which is the extinguished response. There are no even the amplitude here. It means that the retina couldn't function anymore, and it could be this type of wave indicates cases like advanced cases of retinitis pigmentosa, complete retinal detachment, retinal aplasia, and others. This is the normal wave. So this test have its own protocol and procedures. So there are two types of conditions for this test. First is the scotopic, and second is the photopic. So for the scotopic, our eyes will be dark adapted. So before the test, after being given the eye drop, the patient will have to go into a dark room so that the eye could be dark adapted. So when our eye is dark adapted, our rod cells will be the one that function well, not the cone cell. The rod cells itself are highly sensitive to light, and it function in night vision. And there are many rod cells when compared to the cone cells. As for the photopic condition, our eyes will be light adapted. So after the test in the scotopic condition, the test will be done in the photopic conditions where our eyes will be light adapted for I think about 10 minutes, and then the test will be done. So in the photopic condition, as we can see, there is a small amplitude wave when compared to the scotopic, and it also has a short latency. And here, our cones will be the one that function most when compared to the rod cells. As we can see here, it has a smaller amplitude and smaller latency. It is because the amount of cones are way much more less than the rod cells. There are like 6 million cone cells, while there are 120 million rod cells, and that is why the voltage generated when in the photopic condition will be smaller. As for the latency, the cone cells could react more faster than the rod cells, so it has a smaller latency. Latency is the time between the first stimulus, the onset of the stimulus, and the time when our eyes react. So the first condition is the scotopic rod response. Usually our eyes will be flashed with dim white light or blue light in interval
One
of
more
between
minute.
2 seconds. Okay. I'll just so this is the type of test. Angelique.
Application. So the ERG test is taken to determine whether someone has inherited or acquired a disease of the retina. For example, as mentioned before, there is retinitis pigmentosa, which is the loss of peripheral and night vision. We can see the graph on the picture. On the picture, it is a comparison between the normal eyes and the person having RP. So retinitis pigmentosa is caused by damage in the photoreceptor layer, which is the root and cone cells. As a result, there is a decrease of A wave and B wave amplitudes. So the waveform is more or less almost flat compared to the waveform produced by the normal eye. And that is all from us. Thank you.
Okay, thank you, Grace and Anjali. I have a short question. Do you think it can? Do you think it can be used also to detect, for example, color blindness?
Yeah.
It is possible.
Yes.
Yeah,
But
because
I do not know. Aku nggak tahu kayak wave-nya bakal kayak gimana. But as we can see, the theory said that the A wave indicates the photoreceptor, which means the cone and the rod cells. Yeah, and the cone and rod cells are the cone cell, especially, are the ones responsible for our vision, for our color vision, gitu.
Okay.
Second
question is, this is to measure actually the electrical activity at the retina, but as I can see from the first slide, that the electrode is put in the eyelid, rather than at the back end of the retina. So how can it still, how sensitive or how accurate is the measurement?
How accurate?
So you see here that it is in the eyelid, instead of at the really back. Retina is at the back, right? If you learn from anatomy, it's at the back of the eye. So how would it correctly measure the potential at the retina, which is at the back of the eye?
Because it measures. Because it measures the electrical activity that goes back and forth from the retina. Retina is at the back, right? But the retina actually, when we receive light, it actually reflects back. So gimana jelasinnya? Retina is at the very back, but when it receives stimulus, the stimulus is actually reflected back almost to the front, where the optical fiber is located, which is the connection between the eye and the brain. So that connection, the optical fiber, is not actually at the back, but at the front, which is the place that is reflected back by the retina. So the measurement is taken on the flow near the optical fiber, because if there's something like that.
Okay, thank you, Group 2, for your very interesting presentation. So let's move on to Group 1. One moment. Yeah.
Sir, before we start, ini 10 menit kan ya?
Yes.
Okay. Good morning, everyone. So today we will be discussing about a unique device that can detect emotion called EQ radio.
Okay, so EQ radio is an emotion recognition system that relies purely on wireless signals. It is similar to ECG, but EQ radio operates by transmitting an RF signal and capturing its reflections of person's body. It then analyzes these reflections to infer person's emotional state. It classifies person emotional states according to known arousal valence model into one of four basic emotions, which is anger, sadness, joy, and pleasure. And as you can see the picture, the upper picture is the device, and for the lower one is the signal produced by EQ radio, the blue one, and ECG, the red one.
And how does it works? How does EQ radio works? So as we can see in the first picture, the device is put in front of the man. So the device has the first component, which is called FNCW radio that transmit RF signals. And after the signals is colliding to the person's body, it will reflex back. And then after that, the signals will be caught back by the device. And then the resulting RF reflection signals will be passed to the bit extraction algorithm. As we can see in the lower picture, the bit extraction algorithm will separate the heartbeat and also the respiration signals, and it will make segments so that the signals can be analyzed more accurately. And after that, after the signals is segmented, then the heartbeat and the breathing patterns from RF reflection will be passed to the emotion classification subsystem based on the literature. So the device can determine whether the person is angry or sad or happy or like that. And this device also have advantages and disadvantages, but first I will discuss about the advantages. The first disadvantage that this device have is that the RF signals are modulated by breathing and heartbeat signals. So the heartbeat and breathing signals is piled up to each other, so it makes it difficult to read to analyze the signals. But the authors already overcome this problem by use system operate over multiple seconds. So just for example, they just use 30 seconds in frequency domain to just plot the signals. And then the second disadvantage is that the heartbeat of the RF signals lack of sharp peaks which characterize the ECG signals, and this make it harder to accurately identify beat boundaries. And once again, the author has overcome this problem by making segments within few milliseconds so they can analyze per segment. And after that, this device is also a wireless device, and then not like ECG that needs cable and sensors that attach to the body. So the patient is more comfortable with the use if they use the EQ radio. And then the fourth one is the accuracy of emotion classification is 87% compared to the ECG based system, which is 88.2%. So the difference is not that much, just 1.2%. And we can say that EQ radio is a success. But one disadvantage that this AQ radio have is that it is hard to obtain high quality data for emotion analysis because, yeah, as we know that the RF signals is unique. They don't have sharp peaks, so it's kind of harder to analyze it. Next, we go to the case study.
So the EQ radio evaluation was done by conducting 30 subjects. Specifically, the subjects is asked to evoke a particular emotion by recalling a corresponding memory. Is it like sad or happy memories? She or he may use music or photos to help them to evoke the appropriate memory. In each experiment, the subject reports the emotion she or he felt and the period during which she or he felt um, that emotion. During the experiment, the subject is monitored using EQ radio uh, and a commercial ECG monitor in order to compare it. So the experiment shows that the EQ radio emotion recognition is on par with state-of-the-art ECG-based system, which require on-body sensors. Specifically, if the system is trained on each subject separately, the accuracy of emotion classification is 87% in EQ radio and 88.2% in ECG-based system. If one classifier is used for all subjects, the accuracy is 72.3% in EQ radio and 73.2% in the ECG-based system. So for the further development, EQ radio can be used as lie detector, and entertainment industry, like to see how viewers react, or even in daily life when when you feel happy, the music player can play you a happy genre music, etc. And that's all from us. Thank you.
Thank you.
Okay, thank you, Ezra and Andy. One thing, yeah, one main major thing that my critics to you, if a presentation actually to group two as well, before, but I forgot. But in group one, it is more obvious, yeah, because you are already presenting specific result, right? And we know that these are not your work, right? You are basically presenting someone else's work. In that way, you have to uh, mention from the beginning whose work you are presenting here. Otherwise, people will think this is your own work, while it is not. Then it will be considered as, what do you call it, plagiarism or something like that. Okay, so yeah, group two as well. You cite your reference, and it is okay to put at the end of the presentation the references that you took your presentation from. Okay, so this goes for group two as well, but group two presented their presentation, their content in more general way, not necessarily with very specific number of specimen, number of sample, etc. But in your case, group one, you are more specific. So this is definitely refer to certain specific work, specific report. Okay. So please do not forget, not only in my subject, also in later subjects, in all subjects, when you present some things which you don't yourself own, yeah, you are not making them yourself. You have to put the reference. You have to cite the reference. Yeah, this is the standard ethics, yeah, in scientific work, yeah, in presentation or written report. Okay, so later when you submit your report in the Google Classroom by tonight, please add the reference. Okay. So can you go to the earlier slide, please? Just so I have some simple questions. Yeah. So how actually does the how does the is the biosignal being captured here? I mean, what is the mechanism here? Because why does it use FMCW, which is frequency modulated continuous wave, and how can it be modulated by the heart electrical activity?
So why do this the author use FMCW is because FMCW radio have advantages to separate different source of motions in the environment. So if they have multiple peoples, they can capture just specific. They can separate like different sources, and that makes the um, it easily to analyze the signals like that. And so after the FMCW radio transmit, they use RF signals. So the RF signals is the radio frequency that oscillates the rate of an alternating electric current or voltage in the electromagnetic fields form. And then after that, it will, if it bound to the body, it will reflect again, and that signals that is reflect is that is being caught by the FMCW radio. But the problem is that the RF signals is like the breathing and the heartbeat is close to the chest, right? But so they have to separate it between these signals. And one thing that they did is that they know that breathing signals have a constant, like slow signals, but the heartbeat is more faster, so they accelerate the signals to get the precise heartbeat signals like that.
Okay. Second question will be, if there are many people there, how would the system differentiate?
From here, they just test one by one person, but maybe that is the further development that they will make. But according to the definition of FMCW radio, as I mentioned before, that it can separating different sources of motions in the environment. So I think they can analyze which signal from which. So yeah, that's my answer for now.
Okay, I think that will be all for today. Yeah, thank you very much, group group one, for the very interesting presentation.
Especially the last slide, yes, sir.
Yeah, thank you very much. And
Yes,
so
sir.